Welcome to Trial Site News Podcast Series. Today we'll be talking with Dr. Ron Brown, who did a reprint from his Medicina paper and wrote an article on trialsitenews.com about outcome reporting bias in COVID-19 mRNA vaccine clinical trials. Now, Dr. Brown of the University of Waterloo has authored over a dozen peer-reviewed articles listed in the U.S. National Library of Medicine of the National Institutes of Health. And so, without further ado, Dr. Brown, welcome.
Thank you.
The pleasure is ours. Now, Doctor, your topic of relative risk reduction versus absolute reduction has caused a lot of chatter on trialsitenews.com. Could you give a brief overview for those in our audience tuning in today about the differences of these two?
Well, the topic of my conversation with you today are relative and absolute risk reduction measures that are used to describe the outcomes of randomized controlled trials. Now, those trials could be either for vaccines or any other intervention or treatment. And the relative risk reduction is a way to summarize how effective or efficacious the treatment or intervention that's being examined is. Then let me explain. Efficacious or efficacy means how effective is the treatment under experimental conditions. So experimental conditions are very different from real life conditions that you might find in an observational study. A randomized controlled trial is the gold standard for the level of evidence because it has the ability to balance out or control what we call confounding factors. Confounding factors are factors that are giving you the same result as the result that you're looking for, but for different reasons. And the reason why randomization can control this is because, let's say you have known confounding factors and unknown confounding factors among all the participants in your group. If you randomize the group between two groups, then those confounding factors are equally distributed between the two groups. And therefore, what emerges in your results is nothing to do with the confounding factors, but the difference that emerges will be from the treatment itself. And that's why a randomized controlled trial is so valuable. So relative risk reductions and absolute risk reductions are, as I said, measures to report the outcomes of these randomized controlled trials. Now, relative risk reduction measures are also called vaccine efficacy. So in vaccine trials, when you see the word vaccine efficacy, it means relative risk reduction. What's not reported often are the absolute risk reductions. And that's important because absolute risk reductions are more relevant to clinical needs and purposes and applications, to public health applications, whereas relative risk reductions are used for research purposes by researchers. So the relative risk reductions by themselves are not going to give you the information that people, the public, or the physicians, clinicians, or even the public health authorities need to decide, well, how effective or efficacious is this treatment really? And there can be a wide difference between the relative risk reduction and the absolute risk reduction in the same trial. Now, people need to hear both those numbers in order to come to the proper conclusion about the efficacy of the treatment. If you're only getting one number, you're missing the important information, and that could mislead you. And this is a common problem in research today. Many healthcare professionals and the public don't understand the difference between these two measures.
Now, does the FDA require absolute risk reduction? And if so, why do companies rarely use it?
Well, you know, this is interesting because the FDA has published guidelines saying that when research information is disseminated to the public, communicated to the public, absolute risk reductions should be included with relative risk reductions. It's there right in their guidelines because they understand the problem that I was just trying to describe. Now I can get into the details as to why the calculation of the absolute risk reduction is so much different than the relative risk reduction. And to do that, I would have to get into a little bit more detail to describe exactly how a randomized trial for a vaccine is set up. Who's in which group, and how do we count the infections and the efficacy of the vaccine?
Absolutely. Please go ahead.
Well, there's a figure I have in one of my publications that shows you just this. Let me get it up here so I have something to guide myself with. Okay, so in this figure, which you can post while people are listening to my voice, we see two groups, a vaccine group and a placebo group, and we see that there are 100 people in each group represented by these gray figures. And among these gray figures are these red figures called events. In this case, the event is an infection incidence. So in this case, the infection is SARS-CoV-2. More specifically, for the COVID-19 vaccines, they're infections that are symptomatic. So there's at least one clinical symptom with the infection. Now, incidence means that all of the people in the group started out without an infection. So as during the course of the trial, as one person develops an infection, that's known as an incidence. So what we do is we calculate and record the incidence rate of the infection, the event that we're looking at in each of the groups, the vaccine group and the placebo group. Now, it turns out that the vaccine group has another name. It's called the experimental group. And so the rate of the incident in that vaccine group is called the experimental event rate. And the same for the placebo group. The placebo group is called the control group. And so the events that occur in that group are called the control event rate. So the control event rate is the acronym is CER. The experimental event rate is EER. Now, I realize I'm using a little bit of jargon here, but that's why I want you to look at the figure so you can refer back to this and study it a little bit more so that this information becomes more familiar to you. So in the example that we see in this figure, in the vaccine group, we have one red event, one infection incidence. So we have 1 out of 100 people. That's a 1% risk of the infection. We have an experimental event rate, an EER of 1%. In the placebo group, we have 2 infection incidences, 2 events. So that means we have a 2% risk. The control event rate, the CER, is 2%. So now we know the EER, the experimental event rate, is 1%, and the control event rate, the CER, is 2%. Now we can calculate our absolute risk reduction and our relative risk reduction. And you see that on the right side of the figure. The absolute risk reduction is the vaccine risk reduction of the disease. And we calculate that by subtracting the EER, the experimental rate in the vaccine group, from the CER, the control event rate in the placebo group. That gives us the ARR, which is the absolute risk reduction. So in this example, we're subtracting the 1% experimental event rate in the vaccine group from the 2% control event rate, the CER. So 1% subtracted from 2%, it equals 1%. There's your absolute risk reduction. So this vaccine, in this particular example, was effective for clinical purposes to reduce your risk of an event, the infection, by 1%. Now, that doesn't sound too impressive until we consider the relative risk reduction. We use the same exact numbers to calculate the relative risk reduction. The relative risk reduction is supposed to represent the vaccine risk reduction relative to the group that didn't get the vaccine. That's the whole idea behind it. So if we know the reduction in the infection in the vaccine group, which is the ARR, the absolute risk reduction, which we just calculated, we divide that by, in other words, it's relative to the placebo group. And the risk in the placebo group, or the CER, the control event rate. So in other words, the absolute risk reduction, as you can, and all this is on the figure, so you can follow along as I'm saying this, we take the absolute risk reduction, which we already calculated is 1%, divided by the CER, the control event rate of 2%, and that gives us a relative risk reduction of 50%. 50% compared to an absolute risk reduction of 1%. This shows you the power of the relative risk reduction. And it shows you how, if you only had that relative risk reduction number to determine how effective, or whether or not you wanted to use this treatment, without the absolute risk reduction, you would come to, let's face it, a biased conclusion. And I call this an outcome reporting bias. It's biased because you're not getting both numbers, which, by the way, as you recall, I already said the FDA had recommended needed to be conveyed to people when you're disseminating this information to the public. So why have the relative risk reduction at all? The reason we have that is because it makes a good way to quickly summarize in this case, the vaccine efficacy between different trials. That's useful for researchers who are quickly want a comparison between trials. However, it is not useful for clinicians and the public health experts and politicians and the people in general who all need to determine how effective this will be in reality for reducing their risk. In that case, the relative risk reduction is not effective. It doesn't mean that you shouldn't disseminate that information, but you have to also include the absolute risk reduction.
Is it too late then to retroactively look at this data?
Well, as long as you have the data, you can always go back and calculate the absolute risk reduction from the original published data. So no, it's never too late to go back and find it. So if there's a treatment out there, let's just say there's a vaccine for COVID-19, and we're told it has a 95% vaccine efficacy. Remember, vaccine efficacy is the relative risk reduction. So it has a 95% relative risk reduction. Well, what would the absolute risk reduction for that COVID-19 vaccine be? We're not told. So what do? Well, we have the option to calculate it ourselves. And how do we do that? Well, the data is published, and we can find that published data online, and do the exact same calculations that I just did, showing you in the figure.
So, Dr. Brown, if you could, what risks are associated with the vaccines given the participant nor the public is aware of this?
Well, if you're talking about the safety of the vaccine, there are many issues, and we need long-term studies to find out what the long-term risks are. Short-term risks can be very different from long-term risks. My research has not really investigated risks. My research is more in the vaccine efficacy. So there are two issues with vaccines. How efficacious are they? And what are the risks, the adverse events associated with them? So my information is more on the efficacy.
I want to talk to you about an article that you wrote titled Public Health Lessons Learned from Biases in Coronavirus Mortality Overestimation. Now, your goal, it seemed, in this article was to critically appraise the coronavirus mortality estimation presented to Congress. Can you expound on that?
Well, this is important. A year ago, from the time that we're doing this interview in March of 2021. So a year ago, March 11th, 2020, the US Congress heard testimony from Dr. Anthony Fauci about how serious the threat of the coronavirus was to us. And Dr. Fauci suggested that it was 10 times more dangerous, the mortality, more lethal than influenza. He was the one that compared it to influenza. And I believed that at first. But then he went out into the public and started saying on public shows that if we should overreact, if we're overreacting to, in our response to the danger of this virus, it's a good thing. And I thought, that's a red flag. That doesn't make any sense to me. When is overreacting ever the proper thing to do? Can you imagine if our military overreacted as part of their strategy? There'd be no life left on Earth, right? But why is it that this authority figure that we're trusting is going around telling people that it's good to overreact? So that was a warning sign to me. So I started to investigate, well, what are the actual statistics that Dr. Fauci is using here. Now, I have skills as a researcher to do that type of critical appraisal that you mentioned. I can dig back into the research literature and pull out the numbers. Just as you said before, can we always go back and look at those numbers? Yes, those numbers are still there, and we can reinterpret them or critically appraise them. And that's exactly what I did. And I discovered a lot of interesting things. First of all, I discovered an editorial written by Dr. Fauci, co-authored with two other people, where he said that the infection fatality rate, the case fatality rate of influenza was 0.1%. Well, it turns out that that's wrong. The case fatality rate of influenza, depending upon which influenza you're looking at, is about 2 to 3%. For example, that's what it was in 1918. The infection fatality rate of influenza is 0.1%. Now, this whole issue of confusing infection fatality rates and case fatality rates is a serious problem, especially if you're going to do an analysis like Dr. Fauci did and come to the wrong conclusion, and then stir up fear to help get people all riled up about the seriousness of this infection. So all cases are infections, but not all infections are cases. Now think about that for a second. So at the beginning of an outbreak, we're most attracted, or our attention is directed to cases, because those are the people that are ill the most, who are seriously ill, and those are the people who are dying. So that's where all of our attention is focused. So we keep track of those deaths, and we call that the case fatality rate. But in the meantime, there are other people who aren't as ill, or who have the infection aren't ill at all. They're asymptomatic. So those people are said to be part of the infection fatality rate. So you can see there's a difference between the two. You have to be careful when you're comparing fatality rates. The infection fatality rate includes the case fatality rates, but that means it's a lower rate, because now you're including people who aren't dying, who aren't as ill, and who may not even be ill at all, really. Now, when Dr. Fauci was giving his testimony, he tried to adjust for this by taking the case fatality rate of the coronavirus, known at that time from China as 2 to 3%, and then he lowered it to 1%. Why did he lower it to 1%? He said it was because to take into account the fact that we have people, who might have the infection but may not be as sick. Okay, that's fair. But why 1%? Where did you get the 1% from? Why not, 0.5%? Why not 0.1%? He never justified why he used that 1%. And then he compared that 1% modified case fatality rate to the 0.1% fatality rate of the influenza. And he said, see, it is 10 times more deadly. Well, he shouldn't have said it that way. If he was just using a number that he made up with that 1%, it's okay to speculate, but then you can't make a declarative statement and say that it is 10 times more deadly than the flu, which has a 0.1 infection fatality rate. You have to say, well, perhaps it might have, be 10 times more deadly, but he didn't say it that way. He said it directly. Now, if I were to write an article for peer review, and I make a declarative statement like that, and I don't back it up with, true evidence-based statistics, I will never get any of my articles published. And here is Dr. Fauci going out in public, picking a number out of thin air and then making a declarative statement that this is 10 times more deadly than the flu. And in fact, he already confused what a case fatality rate was with an infection fatality rate in his editorial in the New England Journal of Medicine. So these are the red flags that started to provoke me to dig down deeper into this issue of what is the actual lethality of the coronavirus. And my conclusion that I published in my article, that first article that you talked about, was that it's nowhere near as lethal as was originally disseminated to us.
So could you then talk about what are the real world implications for the manifestation of results in actions based on these biases?
Well, we have a lockdown to try to control a fear-based through a fear-based public health campaign, a virus that is not even as deadly as it's claimed to be. I don't think it's as deadly if you look at the numbers, right? And what is the collateral damage from those lockdowns? When I first wrote my article a year ago, I predicted there would be a lot of collateral damage. And I was kind of taking, a risk there because it hadn't actually happened yet. I think it's safe to say that we can look back now a year later and say that it has all come to pass. So those are the types of problems, based upon a fear-based campaign run by, well, certain people. I hate to, just single out one person, because the World Health Organization is involved in this also. The World Health Organization looked at the lockdowns that China implemented, and they saw that the case rates had practically disappeared while those lockdowns were occurring. But that doesn't mean that the case rates, that the lockdown had anything to do with lowering the case rates. There are other issues, and I have another manuscript under peer review right now that explores how the case definitions can change the case rates. China has very different case definitions than we have. And that's all I can say at this point now. I don't really want to get more into it. So there are a lot of issues to still investigate as to how this whole pandemic evolved and why we're still enmeshed in it. This was only supposed to take 2 weeks when it first started. Oh, this lockdown will just be 2 weeks and then we'll be done with it. Something's terribly wrong with this. And there's another issue too, and that has to do with the modern technology we have of genomic sequencing. This only started about 15 years ago, and we have trillions of different life forms on Earth that all can be, sequenced, have a genomic sequence of their genetic makeup. So as we're just beginning to see what the genomic sequences are of these viruses, we're starting to panic. We're starting to think, well, we never saw this before. It must be a new virus. We don't know for sure if it's a new virus or not. We haven't had the proper vetting by epidemiologists to take the information coming from the genome sequencing to vet it to see, is there really a risk here or not? Instead, that information is bypassing the vetting from the epidemiologists like John Ioannidis, for example, and going directly to the public health experts and the politicians who are saying, the sky's falling, we better do our lockdown just the way China did. So there are a lot of issues here that still need to be investigated. And I've been very active over the whole of the past year doing exactly that. And I have several more manuscripts under peer review that are going to try to bring some clarity to how all of these biases have led to these problems. You have to understand that public health officials and politicians have a job to do, and their job is to protect the public. They have to do something, even if they have no idea what they're doing. They can't afford to just be accused of sitting around and doing nothing. That's the end of their job. So you have to balance out how much of what they're doing is really to protect their job and how much is really to protect our interests. We seem to be very anxious to bring, to subject people to all this collateral damage. And not only that, to give up our rights and our freedoms, just the way they do in communist China. So we need to really look more seriously into these issues.
What are some lessons learned or best practices that researchers can take away from your article?
Well, one of them is we need to have open debates about all of this. This is another casualty of the lockdowns. Right now, if you're an academic researcher and you have differences of opinions, which is normally, that's the way normally things go in science. People think, oh, it's science, that science means just one point of view. That's not true at all. We need debates. We need academic debates. There are always opposing views in science. So when somebody says, well, you should believe me because it's science, you should ask, well, which side of the scientific debate are you on, right? Because there's always going to be a debate. And yet we're not allowed to debate anymore. And part of that reason is because of the control by the public health experts and the politicians who can't afford to have a debate. They can't afford that. That would weaken their control over the types of interventions they're trying to impose.
Now, before I let you go today, I want to circle back to the Medicina paper that you published as part of the special issue of the pandemic outbreak of coronavirus. And in it, your paper your paper titled Outcome Reporting Bias in COVID-19 Vaccine Clinical Trials, where you wrote about how this outcome reporting bias would mislead and distort the public's interpretation of the COVID-19 mRNA vaccine efficacy and violate the ethical and legal obligations of informed consent. Now, for our audience, can you expound on why this is such a big deal?
Well, a year ago, I first heard that we have to lock everything down because we don't have vaccines. So that's when it occurred to me, aha, that's what this is all leading to. This would be a good opportunity to show the public in real time how vaccines are developed, how they're appraised, and how the outcomes of the vaccine trials are reported, and how the reporting of the trials can be biased, and how that, as you said already, can violate, the right and the ethical issues of informed consent. And sure enough, that's exactly what turned out. As soon as all of the vaccine information became available from the trials, the last article was published in the New England Journal of Medicine on December 30th, 2020 for the Moderna vaccine. I had all the information I needed now to do the same exact calculations that I had already talked to your audience about when I was describing the EER and the CER, right? Because now I knew how many people were in each group and how many of them came down with the infection. So I could calculate the experimental event rate in the vaccine group and the control event rate in the placebo group. And once I did that, I could calculate the absolute risk reduction, which turned out to be 0.7% in the Pfizer vaccine and 1.1% in the Moderna vaccine. Now, it's true that the 95% relative risk reduction in the Pfizer vaccine and the 94.1% relative risk reduction in the Moderna vaccine are the vaccine efficacy numbers reported in the press. That's absolutely true. Nobody's doubting that. But what's not reported are the absolute risk reduction numbers, 0.7% and 1.1% for the Pfizer vaccine and the Moderna vaccine. Now, some people might say, but Ron, even a 1% reduction over 1 million people is a lot of people. That would be like 10,000 people right there just for 1 million. And I would respond, okay, in that case, then tell people that it's 1% and let them make up their mind. See, this is not about telling people whether or not they should have the vaccine. This is about giving people all the information they need to make up their own mind about whether this is worth their while. And in addition, remember, I said the FDA had put out guidelines that you should report both these, measurements when you're reporting to the public. Well, obviously, the FDA also conducted did the authorization for these vaccines, and they never revealed the absolute risk reduction. They didn't follow this FDA advisory committee didn't follow the FDA guidelines. Right there, this has to come out. And right now, my article in Medicina it's about the most popular article in the journal, and it's gaining popularity. And thanks to the chance to talk to the public through forums like yours, hopefully this information will get out there and people will start demanding answers to these questions. And by the way, Dr. Fauci knows exactly what the difference between a relative risk reduction and an absolute risk reduction is. And I have proof of that. He was actually reminiscing about the 1980 trials for HIV. And he was talking about AZT, which was the drug that was being evaluated in a controlled trial. And he was talking about the absolute risk reduction of how the drug could reduce mortality compared to the placebo. But it was only a 3% absolute risk reduction. It was 8% and 11%. So 8% reduced mortality in the AZT trial, 11% in placebo, an absolute risk reduction of 3%. He didn't call it an absolute risk reduction, but he said it's not statistically significant because it's only 3%. And then he said something really interesting. He said, but the data needs to be further analyzed. And I thought, what's he talking about? He's talking about taking the absolute risk reduction and changing it to the relative risk reduction. This is the way you do vaccine efficacy trials or any efficacy trials for any randomized controlled trial. This is the way, the standard way. It's been done for decades. So he knows it. He should be asked if he knows it, under oath, preferably. And this problem has been going on for decades. If we have time, I got to tell you one more thing. Yeah. Just this morning, I received back a rejection letter from a manuscript I had submitted to a journal on infectious diseases. And the comments were, well, we find your article very interesting. And what I did was I took the influenza vaccines and I analyzed, critically appraised the absolute risk reduction of the influenza vaccines for the past 10 years. And I found that they were nowhere near the 60%, 40 to 60% relative risk reduction. But the comments I got back from the reviewers were, well, we know this already. Don't forget, these are researchers. They're not clinicians, right? And they were saying, well, we know this already. You're not contributing anything we don't already know. Thank you very much. And then I thought, well, wait a minute. You know it. You use these numbers. You're the researchers, but the clinicians don't know it. The doctors don't know it. The nurses don't know it. The frontline workers don't know it. The public health people don't know it. The politicians don't know it. And the public doesn't know it. Who's going to be responsible for that? And that's why I thought this would be a good opportunity during this pandemic to bring all this information.
Well, Doctor, thank you so much for taking the time to speak with us. We really appreciate your time.
Thank you very much.